Good morning, students. Um, in this part of the lecture, we will be covering about the micronutrients. And in the last um, presentation, we saw that the major components of the body includes the carbon, hydrogen, oxygen, nitrogen, and sulfur. So these things are obtained either through consumption of biomolecules such as the carbohydrates or proteins or fats, or due to the intake of water, whereas there are nutritionally important minerals which we spoke about, and these are the macro minerals and such as calcium, phosphate, um, magnesium, sodium, potassium, chloride, which we require in specific amounts in a diet. That's why we call, and these are the we need it in larger amounts, and that's why they are called as macro minerals. Whereas there are also trace elements such as copper, iodine, uh, fluoride, uh, the cobalt, iron, manganese, molybdenum, selenium, zinc, which are needed in lesser amounts as compared to the macro minerals. So these most of these minerals are transported and stored, and they require specific binding to the carrier proteins. So there are various proteins such as the transferrin, which is involved in the transport of iron, chromium, manganese, and zinc, whereas the copper and zinc transport happens through the albumin. The amino acids are responsible for transport of iron and copper, whereas trans, trans-cobaltamine is required for transport of cobalt, and manganese is transported through the globulins. So these minerals are the normal routes of excretion of these trace elements are through either bile, while the digestion takes place because the bile is secreted, and in that secretions you excrete the copper, manganese, chromium, and zinc. Whereas through urine, the chromium, cobalt, molybdenum, and zinc are excreted out, whereas the pancreatic lipases or the pancreatic juices has zinc through which it is excreted out. Through the sweat, we have zinc and the chlorine, which chlorides which goes out, whereas through the mucosal cell sloughing, we have iron and the zinc which is excreted out. So these trace elements, they are inorganic atoms or molecules. So these cannot be digested, and they are required in amounts less than 100 mg per day, whereas the macro minerals are required in amounts more than 100 mg per day. So the bioavailability can be influenced by various factors such as the genetics, the nutritional status, the the nutrient interactions, aging, and all these trace elements are absorbed in the small intestine, mostly the duodenum part, and they are circulated in the blood, and their deficiencies and toxicities are much rarer, except some genetic disorders or environmental exposures wherein large amounts of minerals are coming through water, and on daily basis you get exposed. So the functions, they can serve as cofactors such as metalloenzymes, and they are component of non-enzymatic molecules as well, and they provide structures to the mineralized tissues such as bones. So this is a classic example wherein you can see there is an inactive enzyme, but these minerals can act as cofactors here, and these inactive enzymes combine with these cofactors to form the active enzyme. So once these active enzymes, which are called as metalloenzyme, is formed, the substrate can easily bind to this, and the rate of reaction is enhanced several fold once these metal cofactors bind to the inactive enzymes, and the products are released at much faster rates in this case. So these trace elements are needed in small amounts and are found in the plants and animal tissues, and also the plant mineral content is determined by the soil content of these minerals, and that's why they are very difficult to quantify biochemically. And so the most common nutrient through which the deficiency happens is the iron. So the function of iron um, is the oxygen transport, as you are all aware, and through the hemoglobin, and also it is necessary for the ATP production. So the essential component, the iron is also important part of several enzymes for their function. It also plays important role in the immune function as well as brain function, and the iron deficiency or toxicity is thought to slow down the mental development in the kids. So this is the hemoglobin, and this is the heme, and the iron containing portion of the this is the iron containing portion of this heme. So 70% of the iron in the body is functional and is found in the enzymes and the other molecules. So more than 80% of the iron is found in the red blood cells because there is a hemoglobin in the red blood cells. And 30% of the iron is stored or it is deposited, uh, and it is part of the transport proteins such as the transferrin. So the iron absorption, transport, and storage, and the loss is it is highly regulated in the body. So this is the picture depicting the iron absorption. So the primary regulator of iron homeostasis is 1 to 50% of the iron is absorbed, and if the body needs more iron, it increases the amount of transferrin. So the absorbed iron is carried in the blood by the protein transferrin, and the protein ferritin, it is the one which inside the cells, in intestinal cells, this is stored. The iron is stored in the protein ferritin, and these are the brush border epithelial cells of the intestine. They may either turn away the iron, or once they are absorbed inside, they either stay in the protein ferritin or they gets associated with transferrin and they get transported. So the tissue store or the iron or the incorporate, they can get, once they are transported, they can get incorporated in the hemoglobin, or the red blood cells, they may contain this iron rich hemoglobin, or due to the menstruation they can be removed from the body as well. So once these cells which have the ferritin, the protein ferritin, it can store the iron, but if the cells die, they can slough off and then this can be removed. So the ferritin plays a major role inside the cell for storage of the iron. So the transport happens across the brush border, which I showed in the previous figure, and there is a basolateral membrane. So the heme iron, they can be chemical modification, but maybe it is not required. And the non-heme iron, in case of the non-heme iron, it is reduced to the ferrous form. So there are three forms: the heme iron form, non-heme iron form, and the ferritin form. So this is a picture which is depicting the effect of iron status on the iron absorption. Here is this is the brush border membrane, and this is the intestinal lumen, and these are the basolateral membrane or the submucosal cells, and these are the mucosal cells. You can see in a healthy iron status, the iron absorption requires transport across the brush border and the basolateral membrane, and this gets associated with the transferrin which transport this iron, and these are the square boxes are the ferritin with which inside the cells the iron gets associated. So in case of iron deficiency, the body needs more iron and more transport proteins, so they will enhance the amount of iron transport. So there will be less ferritin will be produced because the body doesn't want it to be stored. It has to be transported to various tissues. So uh, the amount of the transport proteins gets increased, such as the transferrin, result in the increase in the absorption of iron. In case of iron toxicity, however, so when there is too much iron in the body, there will be fewer iron transporters in the on the cell membrane, and there is more ferritin is produced, resulting in the decreased absorption. So what happens is the cells, the iron stays inside the cells and they get associated with the ferritin, and there will be too much of iron which will be accumulated in the cells. So the effect of iron status on iron absorption. So the iron deficiency increase the production of the transport proteins and decrease the ferritin production, whereas excess iron decreases the production of the transport proteins. So here it is a picture showing the iron circulation uptake into the cells and how they are stored. So these are the cells. This is a tissue A, and this is a tissue B, and this is the blood vessel. You can see these cells can take up the iron because there is a presence of the transferrin receptor. So once this transferrin brings the iron, it gets associated with the receptor, and the iron gets into the cell. Whereas there is another tissue, these cells cannot take up the iron because they do not have the transferrin receptors. So the iron storage compounds are either ferritin, which is the main storage form of iron, or the hemosiderin, which is the long-term storage of the iron. So there are various sources which may plants and as well as the animal sources which are rich in the iron, but the decreased absorption is associated with, like I said before, phytates or polyphenols, which is present in the tea or coffee, or the minerals such as the calcium and zinc. So this is a big public health concern in many of the developing nations as well. So whereas the infant, children, and the pregnant and the lactating women are at risk, so the symptoms are they have lower hemoglobin concentration in the blood, the red blood cells also decrease, they have cognitive problems, poor growth, decreased exercise tolerance. So the heme iron is bound to the heme group, and um, the beef, poultry, and the organ meats are the important sources of them. And the heme iron makes up the hemoglobin, myoglobin, and the cytochromes. So the non-heme iron, which comes from mostly the vegetables, the green leafy vegetables, mushrooms, or legumes. So the 85% of this is coming, the dietary iron is coming through this source. So the bioavailability can be influenced by the form, which form they are present, the heme form or the ferric form or ferrous form, or the iron status. So like I said, the enhancers of non-heme iron bioavailability is so the vitamin C and the acidic pH of the stomach can convert the ferric form to the ferrous form. And also the meat factors, the compounds in the meat and poultry or the seafood, so they are also one of the major sources. So there may be chelators, I said, the phytates and the polyphenols, and the major functions include the oxygen transport, hemoglobin in the form of hemoglobin, and they may serve as the myoglobin may serve as the iron reserves, and they play a more important role in the cellular energy metabolism. So this is the hemoglobin is the most abundant protein in the red blood cells. Like we say, see here, there are four protein subunits, four iron containing heme groups here, four, and they deliver the oxygen to the cell and they pick up the carbon dioxide. So whereas myoglobin is found in the muscle, and they have the heme group and the protein subunit. They release oxygen to the cell when needed for ATP production or muscle contraction. So the cytochromes are involved in the cellular energy metabolism. They are involved in heme containing complexes. They function in electron transport chain, and they allow conversion of ADP to ATP. So the iron acts as a cofactor for many enzymes in the electron transport chain, and citric acid cycle, and the gluconeogenesis. So the other roles of iron include the cytochrome P450 enzymes, and they act as cofactors for antioxidant enzymes which protect the DNA from damage, the cell membranes from damage, or proteins from damage. So the cofactors for enzyme to make the DNA as well. So the groups which are at higher risk are mostly infant or growing children or the pregnant women. So the mild iron deficiency can lead to fatigue, the impaired work performance. They can have behavioral abnormalities. They have impaired intellectual abilities. The body temperature regulation can be altered. They can influence the immune system. The severe deficiency of iron may lead to anemia. That means uh, the lower ability to carry the oxygen of the blood. So this leads to the microcytic hypochromic anemia, wherein the cells are pale red blood cells, and they have inability to produce enough heme, they have decreased ability to carry oxygen, and they have decreased ATP synthesis abilities. So, so for the clinical applications, the serum ferritin concentration is about less than 12 uh, micrograms per liter, and you can see the hemoglobin concentrations. For the men, it is 130 grams per liter, whereas for women, it is 120 grams per liter. The hematocrit is for the men around 39%, and for women is less 36%. So the basics of iron supplementation, so they are mostly present in the ferrous iron form. This is the best absorbed form, and there are other forms such as ferrous fumarate, sulfate, and gluconate. So the medicinal, so the most common cause of the iron toxicity when there is too much consumption of iron, and these symptoms may lead to vomiting, diarrhea, constipation, and black stools and death. So the second one, the second mineral, the micro mineral is the copper, and the copper is present in two forms, the cupric and the cuprous forms, and the organ meats, shellfish, whole grain products, mushrooms, nuts, and legumes are the sources. So the bioavailability can be decreased due to the presence of iron or antacids. So the absorption happens in the small intestine and the stomach, and it is influenced by the copper status or the ceruloplasmin, which is the transport protein. And also the excess is incorporated into the bile and can be eliminated in the feces. So the copper function as cofactors for metalloenzymes in the redox reactions. So that for the ATP production such as cytochrome C oxidase, COX, and or the iron metabolism, or the neural functions or antioxidant functions in the form of SOD, the superoxide dismutase, or it is involved in the connective tissue synthesis. The copper deficiency can lead to the hospitalized patients and preterm infants. Antacids can also lead to the deficiency of copper. So the copper functions the essential for normal absorption, transportation, or the mobilization of iron and hemoglobin biosynthesis, and also it's an integral part of COX, and it is stored in most of the tissue, especially in the liver. So the copper deficiency can lead to anemia or depigmentation of the hair or wool. People use the sheep, the black sheep, are as an indicators for testing the copper deficiency, especially due to the loss of the wool crimp is found and leading to the steely wool. So they may have also bone disorders and central nervous system lesions with muscular incoordination. So here we can see the association of copper and dietary sulfur, and you can see the availability can be reduced here, the dietary availability due to the availability of molybdenum or sulfur, the copper absorption can be reduced. So the induced copper deficiency is relatively by high levels of molybdenum and the sulfur, and the sites of interaction is in the rumen, the formation of insoluble copper salts, including the sulfides or the thiomolybdates. So this is in the rumen, this can where the most of the elements the ions can meet, and thereby leading to several interactions. So the net effect of all this is decreased copper absorption. So induced copper toxicity, it is with normal dietary levels of carbon and low levels of the molybdenum and sulfur. When there is low levels of molybdenum and sulfur, the amount of copper can increase and it can accumulate in the liver, and sheep are more susceptible than the other cattle and pigs. So the third micro element is the iodine. As we all know, iodine is very important and for the thyroid hormones, and it is important in the regulation of body temperature, the basal metabolic rate, and reproduction and growth, and it is a regulation uh, so it can be regulated in the body by, or it can be, excess of it can be removed in the urine. So here is the thyroid gland. So people can see during the deficiency there is enlargement of the thyroid gland. So the major source is the iodized salt. So most of the salts are iodized, which provides us enough of iodine. So, iodine deficiency is less. The goiter is a less severe form wherein there is an enlarged thyroid gland, due to which the body attempts to increase the thyroid hormone for production, or there is a cretinism, which is a more severe form, and this is more severe iron deficiency, and during pregnancy leading to serious problems in the baby after birth, such as stunted growth, deaf, mute, and mental retardation. So this is a picture showing the cretinism. You can see the mental retardation and the growth, stunted growth. There is the goiter. You can see this woman having enlarged thyroid gland. So the absorption and metabolism, so the absorbed in the small intestine and the stomach and taken up by the thyroid gland. So the thyroid stimulating hormone regulates its uptake. Here you can see when there is a low blood iodine concentration, then the pituitary gland here increases the release of the thyroid stimulating hormone. Then this thyroid stimulating hormone stimulates the thyroid gland to take up the iodine and produce the thyroid hormones T3 and T4. So the functions of iodine is they serve as component of thyroxine, which is the T4, or triiodothyronine, which is T3, and it regulates the energy metabolism, growth, and development. And the signs of deficiency include lethargy or fatigue. So in 1920s, there was several cases of goiter was observed, and there were campaigns wherein they started the iodized salts to be given to the children. So the iodine toxicity can lead to hypothyroidism or hyperthyroidism and the formation of goiters. So the next element is selenium, and most selenium enters in the blood and it is incorporated into the methionine and makes selenoproteins, and these are stored in the muscles, and the maintenance of selenium is through the excretion in the urine. So the selenium, it's a component of the important enzyme glutathione peroxidase. So it catalyzes the removal of hydrogen peroxide. So the glutathione in the reduced form together with the hydrogen peroxide gives rise to the oxidized form of glutathione and the water, and thereby it reduces the toxicity. So it is also component of iodothyronine 5 deiodinase, which converts the T3 to the T4 to the T3, and it also selenium also improves the killing ability of neutrophils, reduce the prevalence and the severity of mastitis. So it protects the cell from autooxidative damage, which I showed in the previous case, and it shares this role with vitamin E, which is also an important antioxidant. And the deficiencies, especially the white muscle disease in the lambs and calves, the skeletal and the cardiac myopathies are the deficiency symptoms. And also there is a hemorrhagic disease which is found in the chicks. So the selenium content in the soil can determine amount of uptake by the plants and thereby our diets. So the toxicity, so the blind staggers or the alkali disease is due to it causes impairment of the vision in this case. So the selenium deficiency, the important disease is the Kishan disease in China, and this is due to the dietary deficiency of selenium and due to the presence of mutated strain of this Coxsackie virus, which is named after the Kishan County in the northeast part of the China. And due to the toxicity, they gives rise to garlic-like odor in the breath. It leads to the nausea, vomiting, diarrhea, and brittleness of the teeth and the fingernails. So the next is the chromium, and it is found, the content is due to the in the soil, amounts in the present in the soil, mostly the whole grains, fruits, vegetables, and the processed meats, beer are the sources of chromium. So the bioavailability is affected by vitamin C or the acidic medications, antacids. So it is transported in the blood to the liver and it is excreted to the urine and the feces. So the chromium regulates the insulin and the growth and development, and it increases the lean mass in the animals and decreases the fat mass, and it can also serve as the ergogenic aids such as the chromium picolinate. So the deficiency of this may lead to elevated blood glucose levels and decreased insulin sensitivity, weight loss, and the toxicity is very rare, but maybe due to the industrial release of the chromium into the environment. So the manganese, the sources are also the pineapples, nuts, legumes, green leafy vegetables, and less than 10% of it is absorbed by the body. So the excess is incorporated into the bile and excreted in the feces. So the function, they serve as important cofactors of metalloenzymes. It plays important role in the gluconeogenesis, bone formation, energy metabolism, and it serves as a cofactor for superoxide dismutase. So manganese deficiency are rare, but may lead to scaly skin, poor bone formation, growth faltering. And the toxicity can lead to the liver disease or the high water levels in the body. So the molybdenum, the sources are the legumes, grains, and nuts. It is absorbed in the intestine and can be circulated to liver via the blood. Its important function is the redox reactions. It serves as cofactors for several enzymes. It may be involved in metabolism and especially the sulfur containing amino acids and the DNA and RNA. It may be involved in detoxifying drugs in the detoxification of drugs in the liver. So the deficiencies are very rare. Toxicity, no known effects in the humans, but it could disrupts the reproduction part. So the uh, zinc availability can be influenced by phytates or iron or other minerals such as calcium in the body, and due to the acidic substances, the bioavailability can be influenced. It requires proteins to transport zinc into the enterocyte, or uh, it can it requires proteins to bind to zinc within the cells, and the excess of it is excreted in the feces, and there can be also genetic influences which can impact the absorption. So here you can see the acrodermatitis and the acrodermatitis, wherein you can see the skin is getting affected. The zinc deficiency can, with adequate amounts, need to be supplemented in these cases of disease such as this. And in infants, there will be growth failure, and you can see here the skin is scaly, the red and scaly, and it may lead to the diarrhea. It can serve as cofactor for RNA synthesis, and it can stabilize the proteins that regulate the gene expression, such as the zinc fingers. This is an example of zinc fingers that regulate the gene expression, serves as antioxidant as well, and it stabilizes the cell membranes. You can see it can stabilize the cell membranes. The zinc deficiency leads to decreased hepatitis, increased morbidity, decreased growth, skin irritations, whereas toxicity lead to poor immune function, depressed levels of HDLs, and impaired copper status. The fluoride, 99% of them is found in the bones and the teeth. It promotes the mineralization of calcium and phosphate. It inhibits the bacterial growth in the mouth. That's why people use the fluoride toothpaste in order to reduce the bacterial growth and the cavity formation. It is not an essential nutrient, and you can obtain it from potatoes, teas, uh, and the legumes, fish with bones, and people have also the American Dental Association has started uh, providing this fluoride toothpaste where a fluoridation of 1 to 2 ppm is done. So it is absorbed by the small intestine. It can circulate in the blood to the liver and then teeth and the bone, and the excess is excreted in the urine. The function of fluoride is majorly the bone and the teeth health, and it stimulates the maturation of the osteoblast, and it is the topical application decreases the bacteria in the mouth and leading to fewer cavities. So the deficiency not is much known, whereas toxicity can upset the gastrointestinal tract, and it may lead to excessive production of saliva, watery eyes, heart problems, coma. And the dental fluorosis can be seen, and skeletal fluorosis can be observed. Cobalt, since 1930s, it was that wasting disease was associated with cobalt deficiency in plants and soil, and especially they starve for glucose. You can see this animal due to the cobalt deficiency, and the vitamin B12 was found to contain the cobalt. So this is vitamin 12, B12 cobalamin, wherein you need the cobalt in the middle of this. So the cobalt deficiency can be seen in the due to the soils, and the injection of cobalt deficient sheep and cattle with B12 was more effective as feeding cobalt in curing the disease. So it's an essential coenzyme for propionate metabolism. That means methyl malonyl CoA to succinyl CoA, and the DNA biosynthesis and the bacterial synthesis of methionine. So these are the other trace elements. With this, I would like to thank all of you.